Hi all, Nicole Klatt here from the University of Minnesota, and on behalf of my co-chair, Jason Brenchley, who of course we just heard from the National Institute of Health, pleased to introduce our session 2 today on pathogenesis in the microbiome. So we have 4 talks for you. They are pre-recorded for ease of viewing. So I'm just going to introduce all of them, and then we'll let them all play and do questions at the end, and my co-chair Jason will be running those questions. So first we have Eiko Yamada from the University of Colorado that will talk about intestinal microbial communities, and then intestinal microbe, Holdemanella, Biformis, isolated from HIV positive and negative men who have sex with men, increases CCR5 expression on intestinal CD4 T cells. Then we'll go on to Yanhui Cai from Gilead, who will talk about the fecal proteobacteria positively correlating with the activation of circulating ISGs and CD4 T cells in Vesatolimod-treated HIV controllers and ART. And then next will be Shilpa Ray from the Karolinska Institute, talking about alterations in the gut microbiome of HIV-infected patients under antiretroviral therapy. And then finally, James Virga from the National Cancer Institute will close out on a talk by changes in gut microbiome in HIV-infected individuals undergoing long-term antiretroviral therapy receiving Rifaximin, results of the GUTCHEK study. So with no further ado, we will let these videos play and then take all questions at the end. Please feel free to chat any questions you may have. Thank you again for joining us.
Good afternoon, I'm Eiko Yamada from the University of Colorado Anschutz Medical Campus. Thank you for allowing me the opportunity to share my research. Today I'm going to talk about how Biformis, a species of bacteria that is enriched in HIV positive and negative MSM, impacts CCR5 expression on intestinal CD4 T cells. Many studies have reported the gut microbiome of men who have sex with men, MSM, regardless of HIV infection, is compositionally different from that of men who have sex with women, MSW, and women. On the right is unweighted unifrac plot, which shows the segregation between MSM in green to the right and MSW in gold and women in pink, cluster more to the left. As you can see, there is no segregation based on HIV status in the MSM group. Additionally, we and others have shown that HIV-negative MSM have heightened levels of immune activation as assessed by elevated HLA-DR CD38 expression on blood CD8 T cells shown on the left. As you can see, both HIV-negative and positive MSM have elevated percentage of activated T cells compared to MSW. We also examined CCR5 expression on CD4 T cells from colonic biopsies of HIV-negative MSW and MSM, and found that CD4 T cells from HIV-negative MSM have significantly higher levels of activated CCR5 CD4 T cells than MSW, as shown on the right. Since transmission among MSM predominantly occurs during anal intercourse, increased CCR5 expression, the co-receptor for HIV, could increase the risk of HIV transmission. These findings led us to ask the question, can the MSM microbiome increase the expression of CCR5 on CD4 T cells in the gut? To answer that question, we first turned to our in vitro culture assay in which we culture laminar propria mononuclear cells, LPMC, with bacterial communities isolated from stool. Using fecal samples from HIV-negative MSW and HIV-positive and negative MSM as shown in the table, we isolated fecal bacterial communities, FBC, from stool and cultured them with LPMC from resected gut tissue for 24 hours and assessed CCR5 expression on CD4 T cells by flow cytometry. In this experiment, FBCs from HIV positive and negative MSM elevated CCR5 expression on CD4 T cells, especially FBC from HIV positive untreated, compared to those incubated with MSW FBCs. These data suggest the MSM fecal microbiome induces CCR5 expression on laminar propria CD4 T cells. To better assess the impact of the MSM microbiome on CCR5 expression, We turned to a collection of colonic biopsies isolated from HIV-negative MSW and MSM and HIV-positive treated subjects as shown in the table on the left. We had generated single-cell suspension and performed cytoph to assess immune cell subsets and extracted GNA for 16S rDNA sequencing. We then performed linear regression analysis to test for associations between CCR5 and various taxa. As you can see in the right heat map, we found a number of positive associations, many of which are known to be inflammatory bacteria, and one negative association with the genus Bacteroides. The most statistically significant association was with H. biformis, as species enriched in the gut of MSM. To test whether H. biformis is self-induced CCR5, we cultured LPMC with 3 strains of H. biformis for 24 hours and examined CCR5 expression on CD4 T cells. We used H. biformis 80cc reference strain, as well as 2 we had cultured out of our subject. Like with the full MSM stool, we also saw CCR5 upregulation with H. biformis compared to B. uniformis on the left. In addition, H. biformis strain 1 showed higher TNF-alpha to IL-10 ratio compared to B. uniformis significantly. Previously, our group had shown that H. biformis was highly inflammatory compared to other bacteria, as characterized by a high TNF-alpha to IL-10 ratio. It has been shown that both TNF-alpha and IL-10 can regulate CCR5 expression, so we hypothesized the high TNF-alpha to IL-10 ratio induced by H. biformis resulted in the elevated expression of CCR5 on gut CD4 T cells. In order to test if our hypothesis was correct, we cultured LPMC with various ratio of TNF-alpha and IL-10. As you can see in this figure, CD4 T cells in the LPMC cultured with higher ratio of TNF-alpha to IL-10 exhibited higher CCR5 expression, which was tempered by higher levels of IL-10. Taken together, these results suggest the bacteria that induce high ratio of TNF-alpha to IL-10 may increase CCR5 expression on CD4 T cells in the gut. In conclusion, we have found that MSM microbiota increase CCR5 expression on LPCD4 T cells. H. biformis abundance is strongly correlated with CCR5 expression. CCR5 expression on CD4 T cells is dependent on the ratio of TNF-alpha to IL-10. High TNF-alpha to IL-10 ratio increased the expression of CCR5 on gut CD4 T cells. While I didn't have time to talk about this, we have done whole genome sequencing on the 3 strains of H. biformis and found the strain that induced the highest level of CCR5 encoded unique capsular biosynthesis genes in its genome. We are currently following up on this. In summary, these findings describe a novel mechanism potentially linking intestinal dysbiosis in MSM to HIV transmission and mucosal pathogenesis. I'd like to thank all of subject and Palmer and Lisbon lab members. Thank you for listening.
Good morning, everyone. It's my great pleasure to present this data on behalf of the study team regarding microbiome changes in Vesatolimod-treated HIV biological control on ART. Earlier study has shown that administration of a TR7 agonist in combination with a therapeutic vaccine induces CD8 T cell mediated control of SIV in a non-human primate model. Recently, we demonstrated in the phase 1b randomized double-blind placebo-controlled study in HIV control on ART. VES was associated with a modest increase in time to viral rebound as well as a decrease in viral subpoint and intact provider DNA. VES also induced interferon response in a dose-dependent manner as well as increased T cell and NK cell activation. The aim of this study is to characterize the fecal microbiome in HIV virological control on ART and to investigate if microbiome play a role in modulating the VAS treatment outcome. This is the study design. All participants has evidence of virological control for at least 2 years with well-preserved CD4 before the enrollment of the study. They first enrolled on ART for at least 6 months, then received either placebo or VAS with dose range 4 to 8 mg as listed here for 10 doses. VAS was dosed once every other week. After the last dose, all the participants received additional 6 weeks of ART before treatment interruption. The microbiome analysis is to compare fecal microbiome alpha diversity and abundance between people living with HIV and healthy volunteers, particularly address the feature in the HIV virological controller. We also characterize the microbiome changes following Vesatolimod treatment and investigate the relationship between abundance of fecal microbiome and immune biomarkers. At baseline, first we compare the alpha diversity between chronic HIV infection on ART, virological control on ART, and healthy volunteer. This is a small cohort. In our study, we don't see significant difference in the fecal microbiome alpha diversity between people living with HIV and healthy volunteer, but we did notice there's a decline in alpha diversity in HIV infection on ART. When we go down to species level, interestingly, we found the HIV virological control on ART tended to have greater microbiome diversity comparing to other chronic HIV infection on ART. We also compare microbiome abundance at different levels. As others has been reported, we found some families of Bacteroides decline and increased Proteobacteria as well as increased Firmicutes. More importantly, as highlighted with gray background, that for some species, we found only statistic difference between chronic HIV infection on ART and healthy volunteer. And this suggests that HIV virological control on ART present less dysbiosis compared to chronic HIV infection on ART. After treatment, we noticed that VAS partially restored the abundance of some microbiome species to a level that is close to healthy volunteer, with most significantly decrease in Proteobacteria. We also noticed that some species of Bacteroides also restored to a level that is close to healthy volunteer as well. We also perform correlation analysis between the abundance of microbiome and some immune biomarkers by using the data from both treatment arms and both time points. What we notice is that Proteobacteria abundance positive correlated with ISG15, MX1, OAX1, and Ki67 positive CD4 T cell. In conclusion, our data shows that HIV virological control on ART tended to have greater microbiome diversity compared to other chronic HIV infection on ART. VAS partially restore the abundance of some microbiome species, such as Proteobacteria, to a level that is close to that of healthy volunteer. Fecal Proteobacteria abundance potentially reflects systemic immune activation and increased antiviral response in VAS-treated HIV controllers. Further studies are needed to investigate the changes in the pathways and functions of microbiota metabolism that may impact the host immune responses to HIV cure strategy with immune modulators such as Vesatolimod. With that, I would like to address our acknowledgement to our collaborators from multiple institutions. And this is a huge study with efforts from everyone that fully support this study, and especially for those participants and their family and their partners to make this study happen, and particularly they have to spend a lot of time for intensive clinical visit. Thank you for your attention.
Hello and good afternoon, everyone. I'm Shilpa Ray from Karolinska Institute, and I'm really glad for this opportunity to present my work in this forum. So today I'm going to discuss about the alterations in the gut microbiota of HIV-infected patients under antiretroviral therapy. So after the sequencing, we wanted to investigate if there were any changes in the bacterial diversity and the richness in patients after the antiretroviral therapy. And we observed that basically there was no significant changes in the alpha diversity of the microbial communities in the fecal samples of the patients after the treatment. So interestingly, we observed a dramatic change in the gut microbiota composition of patients after antiretroviral therapy, basically at the phyla, family, and the genus level. A total of 139 bacterial genera were identified, and the picture here thereby depicts the taxonomic composition of the microbial communities, which were either depleted or enriched in patients after the antiretroviral therapy. And the overall summary indicates that the bacterial genera of Prevotella, Lactobacillus, Ocelospira, and Bifidobacterium show a significant decrease in the abundance in HIV-1 patients after the therapy. So if there is a decrease in the beneficial bacteria, which is like Lactobacillus and Bifidobacterium in patients after therapy, does this indicate a healthy microbiota? So in search for our answers, we further wanted to investigate if the different treatment regimens had an impact on the alpha diversity of the microbes in patients. And to our surprise, we found that the patients which were treated with non-nucleoside inhibitors showed a significant decrease in the microbial diversity and richness after therapy as compared to the ones which were treated with protease inhibitors. So after looking into the alpha diversity, we were also interested to investigate the effect of different treatment regimens on the gut microbiota composition. So at the family level, we observed that the two treatment groups showed decreased abundance of Prevotellacea and increased abundance of Enterobacteriaceae family in patients treated with the respective ART regimens. And the patients with the protease inhibitor treatment showed an additional decreased abundance of Lactobacilli family after 1 year of effective therapy. So similarly, in the genus level, both the treatment groups show a significantly reduced abundance of Prevotella at the end of 1 year of therapy. And Prevotella is known to be a driver in HIV-induced inflammation. In addition to this, Lachnospira and Ocelospira also show reduced abundance in the patients treated with non-nucleoside inhibitors. An interesting observation other than this was the patients which were treated with protease inhibitors showed a significant depletion in the lactobacillus population after 1 year of therapy. So these overall results indicate that different antiretroviral regimens impact the gut modulation in the HIV infected patients. Hence, to further validate the in silico analysis, we further wanted to investigate the direct effect of antivirals on the gut microbes in vitro. Thereby, from all the 15 antiretrovirals which were experimented in lab, zidovudine showed to have antimicrobial properties against Bacteroides fragilis, Prevotella, and E. coli. Against Bacteroides and Prevotella, the minimum inhibitory concentration of zidovudine was around 4 to 8 microgram per ml. And against E. coli, the MIC of zidovudine was much lower, around 0.5 to 1 microgram per ml. Similarly, efavirenz also showed to have antimicrobial properties against Bacteroides fragilis, Prevotella, and Enterococcus. And the MIC of efavirenz against Prevotella and Enterococcus was around 16 to 32 microgram per ml. And against Bacteroides, it was much higher, around 128 microgram per ml. I would like to mention here that when we are using the concentrations for testing the activity of antiretrovirals on gut microbes in vitro, we are trying to mimic the gut concentrations of the drug, which were actually calculated on formulations based from European Medicines Agency. And these calculations determine that the gut concentrations of the drug are much higher than what is the concentration present in the plasma. The table on the left-hand side of this presentation shows the combination of antiretroviral therapy, which uses efavirenz and zidovudine to have possible antimicrobial effect on their respective microbes. On the right-hand side, the table shows the MIC50 and MIC90 values of zidovudine and efavirenz, which are the values inhibiting the growth of 50% and 90% of the isolates which were tested in this experiment. So with this, we come to the conclusion where I would like to mention that the longitudinal design of this cohort helped us to understand that antiretroviral treatment alone cannot restore the gut microbiome changes in the HIV-infected patients even after 1 year of effective treatment. So more research is required on this topic in order to develop better therapeutic interventions and improved gut health. Secondly, I would like to mention that this study also provides us a proof of concept about the antimicrobial properties of two basic antiretrovirals, which is zidovudine and efavirenz. And these two antiretrovirals are basically used or commonly prescribed in low-income countries. But also in high-income countries like Sweden, zidovudine is used during delivery and during the first weeks of postpartum. So this not only raises the issues about HIV treatment, but also doesn't rule out the possibility that these antiretrovirals can impact the gut microbiota of HIV-infected patients through their antimicrobial property. So with this, I would like to thank Piotr Novak, my supervisor, for his crucial support throughout my postdoc career. I would also like to thank Anders Sonneberry, Ujjwal Nyogi, and the respective groups, as well as Christian Giske and Ashwati Narayanan for their help and support during work. I would also like to thank the funding agencies for supporting our work. And last but not the least, I would like to thank you all for your attention.
Hello, my name is James Virga. I'm a post-baccalaureate research fellow from the NIH in Dr. Maldarelli's laboratory. Today I will be sharing my team's project analyzing the changes in the gut microbiome in a clinical trial of the effects of rifaximin in individuals with HIV undergoing long-term antiretroviral therapy. HIV transmission that persists during chronic infection has a dramatic effect on the gut and is only partly restored by effective antiretroviral therapy. The role of the gut in immune activation remains uncertain. Defects in gut function, such as translocation of bacterial cell products, may contribute to ongoing immune activation present during ART that has been linked to morbidity and all-cause mortality during ART. Other consequences of defects in gut function may include driving low-level HIV persistence. One approach to investigate the role of the gut in HIV pathogenesis in long-term ART has been studying interventions that may alter the gut microbiome with subsequent local and systemic effects. Rifaximin represents a useful intervention. It is a poorly absorbed antibiotic, so its effect is confined to the gut, and it has beneficial effects on the microbiome that have driven its FDA approval for treatment of individuals with IBS with diarrhea, or with systemic complications of dysbiosis such as hepatic encephalopathy. Previously, Tenorio and colleagues demonstrated modest declines in immune activation markers in virally suppressed non-progressors. We investigated the effects of rifaximin in HIV-infected individuals with CD4 counts exceeding 350 cells per microgram. We looked to see if changes in immune activation markers like soluble CD14 could be identified in individuals on long-term ART. We also wanted to measure any changes that rifaximin had with soluble biomarkers as well as on the gut microbiome. We enrolled 42 individuals who had been receiving combination ART for more than 3 years and with HIV viremia of less than 50 copies per milliliter into a randomized double-blind placebo-controlled crossover study shown here. At entry, these participants had a median CD4 cell count of 642 cells per microliter. Individuals were randomized into either the rifaximin group or placebo group for a 4-week period, followed by a 4 to 6 week washout period. Then they switched groups. Data were collected for cellular markers of inflammation from blood samples. Our primary outcome measure was change in the levels of soluble CD14 after rifaximin and placebo. There were no significant changes detected between the rifaximin and placebo phases. We also did not detect differences in the levels of HIV RNA using single copy assays, and we found no changes in soluble biomarkers of immune activation or immune cell subsets. One of our secondary outcome measures was the change in the gut microbiome composition after rifaximin. We collected microbial data from stool samples at the beginning and end of each phase. 19 participants provided samples for all 4 time points. These 19 individuals are characterized by a minimum CD4 count of 350 and at least 3 years on ART. Our study participants were 10% female, 26% non-white, a median age of 49 years old, a median duration of infection of 20 years, and a nadir level CD4 median of 302 cells per microliter. In the overall trial, the entry-level CD4 count was 642 cells per microliter. To measure changes in the microbiome, we first extracted bacterial DNA from each sample. Then we performed 16S ribosomal RNA gene amplicon sequencing. We recovered 25 to 80,000 sequences from 17 participant samples, and 25,000 were randomly selected for further study. Raw Illumina FASTQ files were first demultiplexed using a custom Python script, and then returned pair-ended FASTQ reads were filtered and processed using the QIIME through the NIAID NIH Microbiome Analysis Platform. Wilcoxon signed rank tests were used to compare the differences between the 2 study arms. First, we looked at microbial composition at baseline and found, as have others, that the Firmicutes and Bacteroides bacterial families, dark green and light green respectively, compose 90% of the gut microbiome. Overall, we found a significant decrease in the total number of different taxonomic units after rifaximin treatment by using a log ratio of the day 28 value divided by the day 0 value. It was also significant when measuring the magnitude of the change using delta change, day 28 minus day 0. No specific effects were detected in individual families, including those reported to be associated with inflammation, such as Proteobacteria. We saw a substantial variation in the log ratios of OTUs and their change during the phases, and we were interested in investigating whether any of these changes we saw in soluble or cellular markers of inflammation correlated with changes in OTUs. In an exploratory analysis, we measured the ratio of change in inflammatory marker to the change in OTU for each patient. In general, there are no correlations between the changes in inflammatory markers and changes in OTUs. However, we did detect a significant difference in the change in IFABP2 over OTU. In summary, stool samples provided adequate material for microbiome analysis. We tested the hypothesis that rifaximin will affect microbial composition, reshape the gut microbiome, and reduce bacterial translocation. After seeing no significant changes evident in soluble biomarkers, we analyzed changes in microbial composition. We determined that short-term rifaximin therapy resulted in modest but significant reductions in OTUs. Whatever the changes that occurred were not significant enough to affect the immune activation. We will also be performing a fungal analysis, and these samples are available to study. First, I would like to thank all of our study participants for their time and effort. I would like to thank all my team members within the CRS, especially Dr. Hewick, who is now continuing this research at the University of Tartu, and Dr. Maldarelli, as well as Dr. Ganizan, Dr. McMahon as our site PIs where the study was conducted, Dr. Ute, Catherine Rem, and Sarah Jones, and our colleagues at the NCI Microbiome and Genetics Corps for performing the sequence analysis. Thank you.
All right, well, that was great. Thank you very much to all of the speakers of this abstract-driven session. We're going to open it up for questions now. There are several questions that are loaded, but I'm going to start by taking the session chair prerogative and start with one question for Eiko Yamada. I'm wondering whether or not you have considered using, instead of the bacterial cells as the stimulant to see if it influences expression of CCR5, if instead you just use the spent media, maybe the bacteria are making something that is then influencing expression of CCR5.
Yeah, at first we used fecal bacterial communities to stimulate LPMC and then checked CCR5 upregulation. And then we used 2 kinds of bacteria, H. biformis and B. uniformis. And especially for B. uniformis, we used 3 kinds of strains to stimulate LPMC and checked CCR5 upregulation.
Great, that's great, thank you. But the media that the bacteria are growing in, is it possible that the bacteria are secreting something with their metabolomics that is itself influencing expression of CCR5? So if you don't use the bacteria isolate at all, but the actual media that the bacteria have been growing in.
Oh, so you mean, so what do you mean about media? So we used media just for negative control.
Yeah, but I'm talking about, so when you culture the bacteria, you can take the spent media, because all of the metabolites that the bacteria have been made could be kind of secreted into the media. So if you could take the spent media that the bacteria have been growing in, and then see if the spent media that the bacteria grow in will influence. So it's not the cell, it's something the bacteria is making. It's just something to think about. Great, thank you. And a question for Shilpa from Catherine Luzapone. She was wondering whether or not you separated the patients that had been on antiretrovirals by sexual behavior, and if that might be one of the explanations for some of the dysbiosis, the microbiome dysbiosis that you see.
No, we didn't separate the patients on the basis of sexual orientation, and the focus of the study was basically seeing the differences in different ART regimens, like how they impact the microbiome. So keeping in mind this objective, we didn't consider the gender differences in this study.
Right. Another question for you on whether or not you looked at any protease inhibitor containing ARV regimens.
Yes, we did look at protease inhibitors as well on the 4 bacterial, the microbes that I have mentioned before, and we didn't observe any specific inhibitory activities of these protease inhibitors on the bacteria.
Right. Have you looked at actually any patients who have been on post-exposure prophylaxis? Because then you can kind of remove HIV out of the equation. If somebody that was at risk of getting HIV took post-exposure prophylaxis, and then the only thing that would have been the variable is whether or not they're on the antiretroviral.
No, that's actually a very good point to be noted, but we haven't checked like a patient before getting that. So yeah, but we can consider that.
Yeah, we did do a study in non-human primates where we gave individual ARVs to non-human primates that were SIV uninfected to see if it, and there were some subtle differences in the composition of the microbiome. And then one last question for you, Shilpa, from Satya Dendekar at UC Davis, is whether or not there were any associations with any clinical or virologic outcomes if you measure them in your patients.
After the ART, or is it the correlating with microbiome?
Well, I would think either or, but prior to ARV initiation or after, maybe how quickly the virus comes down after initiation of ARVs, if there were any virological blips, if the patients had higher viral loads before they started ARVs, etc.
Yeah, they usually had like this. So the method part is that we actually use 2 groups of individuals, which were under 2 ART regimens, the non-nucleoside inhibitors and the protease inhibitors along with the nucleoside inhibitor. So there were 16 viromic patients. So this is a longitudinal cohort that we did before and after the treatment. So that is where we try to compare the microbiota composition before and after the treatment therapy.
Thank you. Now we have a question for James Virga from Sergio Serrano Villar. Have you explored the results on the inflammatory biomarkers after stratification by baseline of high versus low microbiota diversity? You're muted. You're muted. So he's wondering if you separate the patients beforehand based upon whether or not they're highly diverse or lower diversity.
We have not separated the patients in that way. That is a good analysis that we should pursue in the future, but we have not separated them based on their baseline diversity.
Right. I had another question for you. When you had a slide where you were looking at the change in IFAB relative to diversity. That went by quite quickly. Did that suggest that rifaximin did in fact reduce intestinal fatty acid binding protein?
What we were aiming to show there was to see any sort of correlations that could be matched between any of the biomarkers with microbial changes. So the best way we thought that we could do that was just to look at the overall taxonomic units that are present and compare them based on changes between their rifaximin phase and their placebo phase. So what we were seeing is that there is some sort of connection between IFAP-P2 and OTUs in terms of how they changed, they decreased during the rifaximin phase. What that means necessarily, that we still need to determine.
Got it. Thank you. Now we have a question for Jenny about the cross-sectional study that you had presented, where people were given compound at different concentrations or not. Were those studies longitudinal analyses, or was it only cross-sectional, comparing patients who had the therapy versus not?
So the healthy volunteers, it's from procure sample. So it's not from the same cohort, but the placebo versus VES are from the control study. Yeah.
So you did have longitudinal samples
Yes.
from for the sample, and so the effects that you were saying were likely attributed to the drug, to the intervention.
Yeah. Mm-hmm.
That's great. And then one last question for Shilpa is whether or not you've studied any patients who have been on second generation integrase inhibitors.
Yes, we have done that, and we have also correlated that with our microbiome data. But since this objective was to see the difference between the non-nucleoside reverse transcriptase and protease inhibitors, so we didn't incorporate that part of the work in here. Yeah.
Great. Cool.
If I can just ask Shilpa a question as well. Did you look at the oral microbiome and see if there's any correlation there at all?
No, we haven't.
It'd be interesting to see if it has an effect as well.
Yeah.
Yeah, no, yeah, for sure. We need to start thinking about all of the ways that each individual.
No, I think it is. I think it's an important, discussion point that it's probably really understudied. So
We are also talking about that, studying this thing also on the forthcoming patients, just to have both samples with us so that we can correlate and analyze, because yeah.
Great. Well, if there are no more questions, I'd like to thank again all of the speakers for phenomenal presentations, and remind everyone that the virtual poster session is ongoing, and please fill out the session evaluation. And like it, love it, hate it, you've got to appreciate that time management with these virtual conferences is much easier than the real thing. I mean, we are right down to the minute we're at time. So thank you, everyone, and see you throughout the rest of the meeting.